In this module, we will focus on identifying genes underlying human traits and phenotypes. The expected learning outcomes for this module include learning how scientists investigate the genome with respect to disease and other phenotypes. We're going to find out how genes underlying common conditions like asthma, diabetes, or obesity are identified. And finally, we're going to discover how scientists find genetic variants that cause specific rare diseases. Now, you may have seen some news stories like these in the popular press. Things like, genes are key to academic success, study shows. Scientists discover over 100 genes linked to autism in largest study to date. And new asthma genes discovered. But how are these genes actually identified by scientists? Let's find out. Scientists use a number of tools to investigate the genome and identify genes underlying human traits. We're going to cover two ways in which scientists accomplish this. The first that we'll cover is by using a technology called DNA microarrays. These particular tools genotype, or in other words, tell us which version of a SNP a DNA sample has at a very large number of locations across the genome. This is a very common method that's used when investigating disorders that are common in the general population and that have a large number of genes contributing to them. If we remember back to our phenotype module, these are things referred to as polygenic traits. A few good examples include asthma, type 2 diabetes, and obesity. The second way scientists might identify genes underlying human traits and disease is by using next-generation sequencing. This is usually used for investigating rare genetic changes that have a very large effect on a specific phenotype or disease. This technique is often used to study disorders that are caused by one gene being monogenic traits or a relatively small number of genes. Let's first cover DNA microarrays a bit more in detail. DNA microarrays, seen here, are special tools that allow the detection of which specific allele or version of a SNP a person's DNA is carrying. It can do this at hundreds of thousands of SNP locations across the genome at once. We see a nice example here of one such SNP. We can see that individuals either carry an A version of this SNP, or the A allele, or a C version of the SNP, or the C allele. The DNA microarray is able to rapidly repeat this detection process at hundreds of thousands of locations across the genome. Now that we have this SNP allele data from DNA microarrays, how do we actually use this information to identify genes' underlying traits? We often use an analytical technique known as a genome-wide association study. The overall goal of these studies is to uncover the genetic basis of a given trait. In these particular studies, DNA microarrays are used to genotype hundreds of thousands to millions of SNPs across the genome. This is usually done in a large population of cases, usually related to some sort of disease measure and healthy controls. You can see a nice rundown of a GWAS over to the right, where we have a number of patients or cases and a number of non-patients or healthy controls. And we then genotype these SNPs in a very large population. We then use algorithms and bioinformatic tools to test SNPs for what's called association, or some sort of statistical relationship with our phenotype of interest. What we're essentially looking for is the increased prevalence of a particular version of one of those SNPs in a large population of cases and controls. Over to the right, you can see that in our cases, a particular SNP has a frequency of 58.3% versus 16.7% in our controls. This tells us that genes in this particular area of the genome may be impacting our phenotype of interest. This has successfully led to the identification of very important genes related to health and disease. Now, why is this actually important? The primary goal of these studies is to better understand the biology of disease. Because genome-wide association studies are hypothesis-free, we're basically taking a large look at the entire genome, it often leads to the discovery of new or novel genes or specific pathways, maybe even genes that we hadn't considered before in the specific disease process. And obviously, by better understanding complex disease and the genes that contribute to those diseases, it may lead to the prevention or even development of new or better treatments. Specific types of data visualization techniques are used to show GWAS results. These are often referred to as Manhattan plots that you see over to the right. These are called Manhattan plots because the association peaks may remind you of looking at the New York City skyline with all of its tall buildings and skyscrapers. You see here an example of a Manhattan plot for asthma. Each dot that you see on the Manhattan plot represents a SNP that we genotyped in that large population of cases and controls. Now, the highest dots that we see, or the tops of those skyscrapers, are the most significant SNPs. And on the x-axis of our Manhattan plot, we see all of the locations along the genome where that DNA microarray measured those SNPs in that large population of cases and controls. Dots reaching a certain significance level, indicated by the dotted line that you see here in the plot, are investigated to figure out how genes close to these SNPs may be functioning in asthma. Let's take a closer look at chromosome 17, our highest skyscraper that we can see on our Manhattan plot. We can see here a representation of all of the human chromosomes. All of the names that you see branching off from the chromosomes are genes that were identified as significant to asthma. You can see that because we look at the entire genome in a genome-wide association study, we can identify a number of genes contributing to asthma in one study. For asthma in particular, there have been over 44 asthma or related trait GWAS that have been currently published. You can see in parentheses, especially when we zoom in on chromosome 17, that often different studies identify the same genes as significant. You can also appreciate that there can be multiple genes in a specific area contributing to a particular disease. Now, our next goal is to determine what these genes actually do and how they might be functioning in asthma. When we take a closer look at the function of these specific genes on chromosome 17, all of them fall into three main functional categories. These include things like functioning of the immune system, mucosal biology, and lung function, all things that make perfect sense when thinking about the asthma phenotype. But remember, because this is a complex trait or a polygenic trait with a number of genes contributing, each of these genes makes a fairly small contribution to the overall disease phenotype. After a genetic location has been found to be associated or significant with a given phenotype, we now have to figure out how these genes are working in asthma. And of course, with complex traits like asthma or obesity or type 2 diabetes, this work is certainly still ongoing. This figure and table includes some great examples of the identification of specific genes using genome-wide association studies in some common disorders, and how specifically discovery of those genes either directly led to the development or use or better understanding of specific treatments and medicines. In general terms, drug targets that are genetically informed have a much higher probability of making it to market as a potential treatment. Let's take the LDL cholesterol trait as an example that you see towards the bottom of our table here. Let's say hypothetically we did our genome-wide association study in a large population of cases, those with high LDL cholesterol, and controls, those with low LDL cholesterol. We would notice that on our Manhattan plot, one of our significance peaks, or one of those skyscrapers, overlapped a gene called the HMGCR gene, which is involved in the process of making cholesterol in the human body. Now, scientists know that a particular type of drug called statins, pravastatin being a very good example, actually inhibit the expression of this gene and in so doing, lowers LDL cholesterol. This obviously reduces the risk for cardiovascular disease and is currently used by about 35 million people across the world to keep their cholesterol in check. And you may be interested to know that genome-wide association studies are still commonly used in research today to learn about specific traits and disorders that impact several genes. In fact, a recent news story about investigating COVID-19 and why certain people have such a severe reaction to the virus as opposed to others is a question that some companies think GWAS can answer. And now let's talk briefly about how scientists investigate rare hereditary disorders. And to do this, we're going to use an example of a very rare disorder called fibrodysplasia ossificans progressiva, or FOP for short. And just a bit of background on the specific disorder, FOP is a disorder in which muscle tissue and connective tissue, such as tendons and ligaments, are gradually replaced by bone, a process called ossification. This is an extremely rare disorder, being diagnosed in only about 900 people worldwide. And so how would we go about identifying the specific genetic variant that causes FOP? And just as an interesting aside, if you are in the Philly area, you can learn a lot more about this and other genetic disorders at the Mutter Museum on 22nd Street in Philadelphia. Remember that we use NGS sequencing to find rare genetic variants with large effects. A quick breakdown of this type of analysis involves sequencing FOP patient DNA using NGS sequencing. This gives us a readout of the sequence of the patient's DNA. We then take this readout and use special programs called DNA sequence aligners to compare it with the reference genome. The reference genome is the genome that was sequenced during the Human Genome Project. So this is often used for comparison when doing these types of analyses. Once the patient's DNA is aligned to the reference, we have special programs called variant callers that can quickly identify where the patient DNA differs from the reference. We can see one such difference circled in red for us. A change from a G allele to an A allele in the patient DNA. We then look closer to identify which genetic changes might be causing the specific disease. For example, for a disorder impacting the formation of bone, like FOP, we may want to start with DNA differences that occur within genes involved in bone formation and growth. When looking at genes involved in bone formation and growth, we notice a single base pair change in a gene called ACVR1. This gene functions in a process known as bone morphogenesis, or in much simpler terms, bone formation. We can see that when we align our FOP DNA to the reference, that the G allele is changed to an A allele, which causes the translation of a histidine amino acid instead of an arginine amino acid residue. This single base pair change causes the bone formation pathway to be hyperactive, leading to increased bone formation and the debilitating FOP disorder.